so far. We're 2 papers behind already. I like that. That's a good thing, by the way. So what we will do, what I think we will do, if it's okay with you guys, is I would like to continue on with our discussion of species concepts, because there are the 2 that are left here, even though we're now supposed to be here, and we'll get to this. We just probably won't cover all of these. The fact of the matter is, why I give all of these papers or readings to you guys in all of these short courses is not so much that we can cover every little detail in them, but so that you read them and you understand, hopefully, the context, sort of the broad context, for example, of species concepts. You need to read all of them in a concerted fashion now, I think, so that you can get a broad understanding, and that helps the discussion, but it also helps, I think, you understand, yeah, that's how this works. You know, and maybe this is how I should think about this. This is maybe how I will be able to apply it to whatever I'm doing. All right, so these 2 species concepts are left, and I would actually like to touch on both of them. And so could someone tell me what this species concept is? What the name of it is? Phylogenetic. And so what do we mean by the phylogenetic species concept, or what did Joel Craycraft mean? If I was going to apply this to a group of organisms that I'm working on, how would I apply the phylogenetic species concept? What would I need to do? Yes?
I think what he was saying, that counting all the morphological characters that you are going to count, and then just build the phylogenetic tree using all of those characters, and then whichever is one clade is one position.
Okay. So, with that in mind, let's go through some thought experiments. So, we have a data set, whatever it happens to be. Maybe it's morphological, maybe it is molecular. Quite often nowadays, in fact, more often than not, we're going to use DNA data to draw phylogenies, okay? And so we use something like Beast or Mr. Bayes or something, and we draw a tree. Okay, and now we have, and we didn't have this tree before. How many species do we have? I'm sorry, I'm driving you nuts, moving back. I always walk back and forth. I'm sorry. This is actually what I do all the time. I'm not doing it just to annoy. So, how many species do we have? Come on, just take a... Come on. I'll just make fun of you for the rest of the time if you get it wrong. No, I mean, how many species do we have? Oh, geez Louise, don't tell me you have to define a cutoff again. I'm going to get so... No, okay. How do you define a cutoff? No, I mean, I'm not making fun of the phylogenetic species concept. I do make fun of my phylogenetic species concept colleagues. I call them terrorists a lot of times, actually, because of the way they act about how they're doing their phylogenetic species concept is so, it's almost like a religion. That I'm making fun of phylogenetic species concept. But so, how many species? How do we do a cutoff here? Why aren't there just 5 species? Let me ask that way. Why can't we just say A, B, C, D, E? There are 5 species. I mean, we can, but you're saying we shouldn't just do that. necessarily.
A and B originate from another parent computation.
Sorry, A and B.
Originate from one point which has diverged from the original branch.
Okay.
Like from the main branch.
Uh-huh.
So
No, you've had clade genetic events. Okay.
so you already have so many branches.
Okay. So are you saying that's a species? Like this? This is a serious question for phylogenetic species concept folks. But why can't we just say there are 5? Your answer is a good one. You know, there are complexities here, but how do we decide how many there are? What do you mean by decide on a cutoff? It is what you have to do, by the way. Amount of divergence? Okay. So we could use maybe amount of, okay, divergence. But is that, people do that, by the way, using this. But is that really phylogenetic species concept? Isn't the phylogenetic species concept simply the ends of each one of those should be a different species? I mean, okay, I know I've read too many books. I've read Willie Hennig's Phylogenetic Systematics. It was translated into English in '66. No, I did not read it in '66, okay? I was 9. I was precocious, but I wasn't that precocious, okay? In his book, he says they need to be monophyletic. There needs to be an ancestor-progeny relationship. But basically, he would have argued, as far as I can tell, that there are 5. Okay. Now we come, and Willy Hennig, I guess it was, would have been using morphology, right? Morphological traits. His book came out in '66 in English. He wrote it before then. So what happens if we have genomics or genetics? We have 100 loci. What happens to this lineage if we sample 100 different individuals for 100 loci, do you think? Do we end up with one lineage? No. No, we end up with something like that, so with 100. Probably not 100, but a lot. So what do we do about that? Where is our cutoff? This goes back to this. Where do we decide when the tips of the tree, the phylogeny, reflect different species, reflect different taxa? How do we do that? This is a majorly applied species concept, quite often in plants, but in animals as well. So this is a major question, yeah.
I think what people do is that knowing that A and B are different species, then if we draw a tree like this using molecular data, then the difference between A and B, that should, because of the closest, then that difference should be the minimum difference that there should be when you're defining the species.
Okay, so it's like a barcoding.
Yeah.
But how do they know A and B are different species?
From other sources.
Okay, so this is great. I'm really actually not trying to embarrass or put you guys on the spot. Do you see the dilemma with every species concept we can come up with? You have to use basically some other species concept to figure out your Bayesian prior, basically. Does that make sense? So you are here saying, well, we know A and B are different species. You know what Willy Hennig said in his Phylogenetic Systematics? Just blew me away, and I've used it as a quote quite often just to irritate my Phylogenetic Systematics I know, it's terrible that I do this. But anyway, the father of phylogenetic systematics, the person that they honor every year by having the Hennig Society meetings. Okay, nothing wrong with that. But anyway, he said, well, the way you know that they're different species, yeah, we draw trees, but they have to be reproductively isolated. I just think that was hilarious. And so I like to quote that occasionally, you know, slide it in there, because the phylogenetic systematics concept, phylogenetic PSC, is that you just use the tree shape. You don't use any other information. Just like the biological species concept is all around reproductive isolation or not. Okay? And so, but both, you actually use other information to infer phylogeny or which part of the phylogeny matters the most. Okay. So I just want to point that out, that this depends, and my people at my colleagues who I know at the American Museum of Natural History, if they could hear this right now, if they run across this, they'll be so angry. It's all tree shape, Mike. We tell you that every time, and it's like, nah, it's not. Now, you had to decide how A and B, your least diverged. You had to make a decision of what was the least divergence that added up to a phylogenetic species, period. And that divergence is not tree shape. It has something to do with something else. There's nothing wrong with that, by the way. I just want to encourage you, as you think through these species concepts, there is nothing wrong with the fact that they have to play off against one another. The only time it becomes a problem is when you're Ernst Mayr and say this is the only way you can measure it, period. Or when you're someone doing PSC and they say any other information is just subjective. You just draw your trees and that's what tells you how many species you have. Okay? So, PSC. Questions, comments? Yes?
Sometimes what we do, we just use some statistical test or some higher approaches to decide whether the number of amount of divergence is significant or not,
Mm-hmm.
to be called as a different species.
Mm-hmm.
Is that really useful or helpful to decide whether the divergence is significant? Statistical test.
So really what you're saying is you're looking at constellations, from what I know and from talking to people and applying this myself, you're using constellation, basically asking what the variance is between A, B, C, D, E, right? You're asking about the level of divergence, but also a sort of a variance around it. How do you choose your amount of variance that's significant enough to be different species?
So for that, you just compare the data of your species complex with the, as she said, if you know what two different species are, the data or amount of the divergence present in between the two well-known different species.
Mm-hmm.
So you compare how much divergence is there in your species complex,
Mm-hmm.
that other species,
Mm-hmm.
and depending on that, and I don't know, I'm just saying that is possible.
Yeah, absolutely. That's exactly what they're actually doing. You're exactly right. It's just that we need to understand that we have a prior. In Bayesian terms, we have a prior, right, which is A and B are different species. We have to have, I mean, this is a scientific method, by the way, Bayesian analysis. I just love it that we call it Bayesian. It's just a scientific method. Priors are the things that we were feeding into to give us our hypotheses all along since scientific method was basically invented. But it's just that we need to understand that it's not just tree shape. It has to have some other information in there before we can statistically determine or whatever we're using. Yeah, but that's exactly actually what people are doing. They're asking the question, they're basically doing a PCA, a principal components analysis in many ways, although it's not a PCA a lot of times, but it's just basically asking if you have different constellations that reflect A, B, C, D, E. Do they make sense? But it's in a phylogenetic terminology. So very powerful, can be very powerful to tell, like I say, I mean, this is what I did for 6 years. And so I have nothing against this. It's just that when we don't understand that actually we have an assumption there, it's when we become radical about it and say it's only tree shape or it's only reproductive isolation or if it's only whatever, and we don't allow the biology to be there and to understand that we have assumptions here that A and B are different species. For some reason, we assume that. And it's probably good reasons, but that's an assumption. Okay, other questions, comments about PSC?
Sorry, it's still not clear how it defines the species using some biology. Sorry, did you say it already? Or?
So, strictly speaking, the PSC argues that you just count the number of tips. The tips, the terminal branches, and you say, in this case, it would be 5. If we're just going to do, strictly speaking, the phylogenetic species concept that Billy Hennig laid out there, that's how you do it.
As many tips, as many number of taxa that you include in your biology. Not even taxa, as many tips as the number of individuals.
That's the problem, right? Because when Billy Hennig was doing this, and before we had the capability or the capacity to do gene sequencing, for example, or to get any kind of particulate kinds of data. Before we had that, we only had quantitative data, and so they overlap broadly, and we could say this, all the morphological data fit here, 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 here. And they're different from one another. We'll count the tips. There were still problems with this, and that could work. The problem is, like I said, if we, and Joel talks about this, Joel Craycraft talks about this in his review, with DNA kinds of data, like you said, the number of samples is probably, if we have a number of loci, a lot of loci, the number of samples is going to equal the number of tips in that tree. Right, I mean, if you just draw, it's going to be a gene tree with 1,000 tips if we have 1,000 samples. And so that's where we go back to what you were talking about, and we ask the question, we have a touchstone. We have something within this that tells us this is the minimum amount of divergence that is necessary for these to be different species. And so if we have a cluster like this, we say that's not enough divergence. There's not enough phylogenetic resolution in the PSC speak. Okay? So we still count tips, but we collapse them if they do not have enough divergence from one another. We collapse them just like we do when there's not enough bootstrap support. We collapse an internode. So we collapse those. That's how but we still, with the phylogenetic species concept, we still go back and count tips. But it's with added information about how different they have to be. Otherwise, it becomes absurd. And that's what Joel pointed out. You could look at, and John Avis has pointed out as well, you could look at some tiny morphological character that varies across individuals before DNA, varies across individuals, and have just an enormous number of tips to the tree there. And that would be absurd. It wouldn't reflect biological reality. But yes, you're right. You could have as many tips as you want if you have highly variable data. So we have to have something else to ground that in. But I'm trying to get you to think about the strict application of the biological species concept, the phylogenetic species concept, okay, before we then say, that can't work exactly that way. This is what we have to do to manipulate it. Yes? How does it? Oh, we're assuming reproductive isolation. No, the phylogenetic species concept, folks, would absolutely not add that one in. Not in strictest terms. It's supposed to be, it's not, tree shape is everything. It's the pattern, evolutionary pattern, not process. Reproductive isolation, I'm just telling you how this works. I'm not agreeing with it. But phylogenetic species concept is based on tree shape, i.e. the number of tips. They're not assuming that those are reproductively isolated. Not if they're applying the PSC in the way it's supposed to be applied. Because that's a biological attribute, and they're just interested in pattern. Oh, sorry. Sorry. Well, it's strictly speaking, yeah, if you're doing phylogenetic species concept and assuming just pattern, yeah. But what we're saying is that that's going to lead us into absurd conclusions where every individual sample is a different species, right? So we have to have some other information. We have to apply some other information if we're going to use this. And there are ways that people do this all the time. I mean, they really do use the PSC to try to, and they add other information like reproductive isolation or whatever. But I'm just saying the strict application of this is number of tips equals numbers of species. And we have to build from what the assumption of the model is and then add to that before we have to understand what the assumptions are underlying it. It's like Ernst Mayer's. If they reproduce or intergress, they're the same species. Well, that would reduce all canids to 2 or 3 species, and nobody's going to do that. Why aren't they? Because they're reproducing. Because nobody's going to apply the biological species concept that strictly. They're going to use other information to inform that decision. Yes, go ahead.
Actually got the answer to my question, but I'll start giving
Okay.
so I was going to ask that wouldn't traits being convergent or could big species really mess this concept up?
Yes.
I guess that you would not be counting those characters.
You wouldn't be necessarily counting those characters. Yeah. You wouldn't necessarily be counting them because of the way that they would put the data into those. That's true. But convergence and parallelism actually are really major problems for phylogenetic reconstruction anyway. I mean, but they just get the estimate of homoplasy. You know, homoplasy covers everything. That is evolutionary noise. They get an estimate of homoplasy when they reconstruct these kinds of gene trees, or if it's good, species trees, whatever it happens to be. Then they get an estimate of homoplasy and they just say that doesn't count. So genetic exchange is a homoplasy. Incomplete lineage sorting is the homoplasy. That sort of thing.
This concept also uses principles such as common ancestry and descent, right?
Suggests.
Also uses principles such as common ancestry and descent.
Yeah. Yeah, absolutely.
So it's not just it's recognizing the tips as different species, but it also takes into consideration the principle of monophily, for example. So what I'm saying is, in a way, it can be very useful. For instance, if you can't if you didn't have the phylogeny, and if you had classified A and C as the same species, but B is a different one. Using a phylogeny to tell you that using a molecular phylogeny to tell you that because A and C are not strictly monophyletic, they can't be the same species.
Yeah. Yeah, go ahead.
In that respect, I think it's a very useful.
Let me also agree with you and say absolutely. And in fact, we will talk at length about how we use phylogenetic incongruence or non-concordance to test for things like genetic exchange. So in other words, morphologically, I think that's what you were saying, that A and C look like they're the same species, but you do molecular and they're not. That's called a discordance, non-concordance, and that tells us something about evolutionary process. Often it tells us that we're looking at incomplete lineage sorting in the molecular versus morphological or genetic exchange. Those are our alternative hypotheses here. But absolutely, phylogenetic information and the progenitor and offspring branches, if you will, that pattern is incredibly important. I would argue it's more important for understanding process rather than numbers of species, but that's me. That's my bias. I'm more interested in character evolution or whatever happens to be. But absolutely, you're exactly right. Okay, what else? Anything else? I would like to, what time are we stopping? Now, well, since we started at a different time, I'm all messed up.
3:30. Stop at 3:30.
For a break. 3:30, perfect. All right, so let's talk about Templeton's paper, chapter. Same book as Joel Craycraft's, as you can see. So what was Templeton's species concept? Remember, if you think of other questions on these other species concepts or anything else we've talked about today, just shout, okay? Tell me about Alan Templeton's species concept. What was the name of it? What did he name it? Did you pick up on that? Let me ask this. Was anybody confused by Alan Templeton's species concept? I was. Took me about 6 readings to get through it and to understand what he was trying to get to. What is the name of Alan's species concept? What did he call it? I do this usually, by the way. I pull this one up. I should have done this already on the screen. It's because I like to go through one of the graphs with you guys. I think that's it. Yep. All right. Let's see. We'll go back. So, name of his species concept, okay? The cohesion species concept. Can anybody tell me what the components, how would Alan define a species or 2 species, or whether he has 4 or 5 species? Suggestions?
Based on multiple lines of evidence.
Okay, absolutely. Multiple lines of evidence. What does he talk about, though, in terms of one of the ways that we can think about it? He compared his concept to what other concepts? Do you remember? what did he compare his concept to? Whoop. And we'll get to what he was talking about. Okay. Let's work through this. His is, okay, why do I give you one that is tough to work through, that's sort of dense, really? And the answer is, this is sort of how I do it. And in fact, this is pretty much the way I think of applying species concepts. And that is that nature is complex, speciation is complex, and we need as much data as possible to try to understand speciation, numbers of species, whatever else we're working on. And so Alan talks about problems for the biological species concept, which he calls isolation and recognition concepts, or flip side of one another. But isolation is the biological species concept. He changed the terms. And he talks about the biological species concept in particular as having problems with organisms with too much or too little sex. Now, what did he mean? I mean, you can see it on this, but what is he talking about there? In terms of applying the biological species concept in particular, what does he mean by too much? He was doing it tongue-in-cheek, obviously, but too much sex or too little sex? Okay, sexual, asexual organisms. But what about their biology makes them difficult or impossible to apply the biological species concept to? Okay, so it has to do with the reproductive isolation, obviously, the biological species concept, reproductive isolation, yes? Okay, too much sex, something called syngamions. Syngamions is a terminology that you see quite often in the plant literature. Lotze actually defined it, I think, in 1902 or something. It's the largest unit of basically reproducing units. And it's often, syngamions are often pictured, if you see them in a figure somewhere, it's often a circle with species names around it. Doesn't have to be species, could be subspecies, could be whatever taxonomic unit. And basically with lines running like that between units that hybridize in nature. And so this is sort of a pictorial, lousy drawing, but a pictorial of a syngamion. So North American canids, European canids, canids in general are a syngamion. You could put, and I'll show you figures, phylogenies with migration or introgression indicated on them of all the canids. And you could draw all the canid species around the outside there, and you would see lines drawn between them. Ancient as well as recent. Bears are the same way. Ovids, cervids, all sorts of mammals are like that. Plants, of course, we can do that too as well. So that's what a syngamion is. So if you have organisms that are actually hybridizing, integressively hybridizing or crossing quite frequently, then Alan argues that you can't apply the biological species concept to them, because if you do, you will reduce them all to the same species. And he argues that may not be appropriate. Maybe, but it may not be. On the other end of this graph or figure that he made, he had asexual taxa. So my question to you is, why can't you apply the biological species concept to asexual taxa? I mean, they're isolated. If they're completely asexual, they're not sexually reproducing with one another. So they're isolated. From a BSc, the BSc would just say we can't apply to it because they don't reproduce sexually. We can't measure sexual reproduction and isolation. So definitionally, they're not able to be included. But Alan Templeton says, yeah, but it's even worse than that, because if they have any asexual reproduction at all, or even strong selfing, that reduces the area that the biological species concept actually covers, is his argument. What does he mean by, so here's his species concept that he said covers all this area, okay, that can actually apply to all this area. What is genetic exchangeability and what is demographic exchangeability? Any ideas? What do you think genetic exchangeability would be? First of all. So he's trying to, remember, we're trying to define, he has two things. He's holding them in his hand, two birds. And he wants to know, they look morphologically different from one another, but he wants to know whether or not they're one or two species. Okay, and he's going to apply his species concept to that. So he wants to know if they're genetically exchangeable. What do you think that refers to? Yeah, we can think of it as being reproductive isolation, but it's more than that, right? It really is genomic compatibility, all sorts of different things like that that he's referring to, that he's putting into that box. And so we can use genomic architecture, we can use amount of divergence, lots of different things that we put into this to estimate now with our organisms, to estimate genetic exchangeability. What about demographic exchangeability? I think this is where he really, I just don't think it's a good terminology, but it's incredibly important. What do you think he means by demographic exchangeability? So genetic exchangeability means if they're completely genetically exchangeable, they're basically down here. Okay? But that's still, in his species concept, does not mean they're not different species. Because maybe they're not demographically exchangeable. But what does he mean by demographically exchangeable? Very well done. Yeah, so it really, the way I would say is exchangeability of realized niches or ecological exchangeability, but he is trying to put some other things in here in terms of demographic. But really, this refers to the environmental perspective or environmental correlates. You can think of it as being when you guys and when we do ecological niche modeling, it gives us an idea of whether or not we're looking at exchangeable units. Where do they occur? How broad does their environment look? Does it broadly overlap with the other form that we're looking at? Okay? What if we have individuals like this, or sorry, lineages like species 1 and species 4, we think they're different species, but they're both genetically and demographically exchangeable. With his species concept, are they different species? No, right? But if they're reproductively or genetically exchangeable, but they're demographically not exchangeable, they live in different niches, then they are, according to his species concept. Likewise, if they are genetically not exchangeable, but they are genetically exchangeable but demographically not exchangeable, they're different species. But if they are demographically exchangeable and genetically not, then they can be considered different species. Do you see? So he's playing this. This is why I say we apply this more in our lab. We still use phylogenies, gene genealogies, really, as a way of depicting it. Or actually, we use networks because we allow migration events between them. And that's, I think, better reflection rather than just trees. But these kinds of information are what we're overlaying on that are giving us an inference, not only about how many species we have, obviously, but really what the species, how they might have evolved, what their adaptations are, etc. Okay? Questions or comments about these? By the way, just to tell you, applying this in strictest terms, I think, is impossible. So the easiest thing to do is just draw a tree and count the tips, honestly. Seriously. Really is. Get an idea of what the level of divergence is that you need and all that sort of thing. Because you're not going to know whether they're reproductively isolated unless they overlap in nature, right? Not really. And, that can give you some information that's easier to do if you're trying to decide about species. But honestly, you can't just draw the tree and count the tips, right? Not if you're really interested in evolutionary pattern and process. That's what I keep telling my phylogenetic species concept people, and they disagree, though. So I just need you to know that what I'm saying goes against what a lot of people do. They just say, no, you don't have to worry about all this. You just do that. They may be right. I don't think so, but they may be right. Okay. Questions or comments over species concepts? Okay. Close that. I'm going to close that down, and I'm going to go ahead to session 2. Aren't we in session 2? And I want to... Someone was asking me, so now what we're doing, let me go back here. Okay, this is what we're going into now, which are modes of speciation that I want us to discuss now. In other words, how species may form. Yes?
According to the last concept, you were talking about an example of wolf and dog. According to that species concept, so that wolf and dog can be differentiated into two different species?
According to Allen's, the cohesion species concept? Yeah, no, according to Allen's, they would not. And so the question is like canids. So in North America, we have gray wolves, red wolves, coyotes. Domestic dogs are mixed in there, I suppose, to some degree. We have some other canids, gray and red fox, which can be considered there too, because they hybridize, integress. So the question is, under Allen's cohesion species concept, what would we do with all of those? The answer is that though they show extensive interaggressive hybridization in very large zones, gray wolves, coyotes, etc., they're definably different in terms of their ecological niches. And also their adaptations, their morphologies that reflect different adaptations. So they're not exchangeable. They're a syngamion, but they're not demographically exchangeable. That's how we would put it. Yes. And we see it's all, remember what we're talking about here, and that's why I say applying any of these is difficult in a case by case. But applying Allen's, no. Let's say applying the biological species concept, is there enough hybridization amongst coyotes and gray wolves and all to actually call them the same species? Some people would say, well, they're not intergressing that much. Well, yes, but according to Meyer, they actually are. In certain areas, we have, like I say, 30% of the individuals are hybrids. That's huge. I mean, that is an enormous amount of migration, integration. But what we can definitely point to is their habitat preferences, their behaviors are definitely different, remain definitely different in terms of what we would call a gray wolf and a coyote. But still, so if we applied the biological species concept, we really would have to reduce them probably the different subspecies. But once again, nobody has ever suggested that for these forms. And there are many, many animals and plants out there that fit that, where there's extensive genetic exchange, interaggressive hybridization. Nobody is pointing to the fact that, yeah, we ought to take these very different ecological and phenotypic forms and merge them into the same taxonomic unit. Because they know they occur in different environments, et cetera. But it's applying these species concepts is always going to be somewhat subjective, right? It is always going to be subjective. And it's coming up more and more with more data we have, it gets worse. The more molecular data, for example, the more genomic information we have, it just gets worse. And I don't mean that we should stop producing genomic data, but the more data points we have, the bigger constellation of gene trees we produce. And the more detection of genomic exchange and genetic exchange we have. So we have to keep modifying our evolutionary models to fit with our observations, rather than saying the observations need to fit the models. Yes?
Bit off topic, but are there any questions which absolutely you need to get to number of species, like for example conservation?
Yeah. So are there questions where you absolutely need the number of species? I would say that the strongest inferences for character evolution come when we have a really well-resolved network. So for example, if we want to know how many times hummingbird pollinated plants, is hummingbird pollination easy to evolve? Or is it not? And does it involve a lot of genes or few genes? Well, we can get inferences. The only way we can get those kinds of inferences is knowing how many lineages we actually have there, and then being able to test character evolution. And that's just one kind of test. So we really do have to have very good phylogenies for that. I would argue very good networks so that we can follow how the genes are moving around, but still we have to have some sort of an idea of where the different characters ended up on that network to be able to, and there are huge numbers of studies like that where people are interested in character evolution, whether it's convergent, whether it's parallel or whatever. So yeah, I think conservation though is one that obviously, depending on where you are in the world, species species have more value. Sorry, taxa being called different species gives it conservation value that subspecies don't have. In fact, you see some things in the scientific literature that make you wonder whether or not they've split just so they can conserve. Yeah.
So, taking the example of creative only, you said like there can be about 20% of incarnation. So, I'm just wondering, like, in spite of having so much of hybridization, how the species are able to maintain their species-specific character over so much
Yeah.
generation?
It's a good question. So how can you have, we actually will talk about this at length even in next, maybe tomorrow. No, 4 and 5. But so how can units that we call different species or different whatever, but we recognize them as being very different from one another, how can they remain different if they are exchanging genes, if they have a lot of introgressive hybridization going on? And the short answer is that what we're coming to understand is that the genome of all organisms are mosaic. Genomes are mosaics. They're mosaics of different bits and pieces of DNA from different sources. Some vertically transmitted, but many horizontally over as they evolved. And what that tells us, I think it gives us an answer to your question and to all of our questions. How do you end up with this kind of biodiversity? And the answer is natural selection. Ecological, probably environmental, strong environmental selection that forces these, continues to have linkage disequilibrium between genes scattered around the genome that are not being transferred, that are keeping these phenotypic units together. Sometimes not. Sometimes you get adaptive trait integration and they start pulling together like Darwin's finches. But you still have phenotypic units, sort of constellations that you say, well, that's Geospiza scandens and that's Fortis, even though their beaks look a little bit alike now. So I would argue that, it's because of that kind of natural selection. That's how you end up still with that kind of strength of, or magnitude of introgressive hybridization, ending up with something we would define as gray wolves and something we would define as coyotes. But let me hasten to add, we also have coming out in North America, there have been a number of studies demonstrating that introgression has also led to new hybrid lineages that are distinctive from the parents. Whether they're different species, we would call them homoploid hybrid species. But all of that is going together. It's not just that they're being held apart by selection, but that novelty is leading to new novelty, like the dog genes in gray wolves. In certain populations, it's adaptive. So that's new novelty in the gray wolves. So this is why I say that every time we look, it gets more complex. I think it gets more interesting, but every time we look, it gets more complex. And what I mean by look, when we start comparing behavior of the gray wolves that are black in Yellowstone, and we wonder why in the world they're black, and we find out it's dog genes, that have introgressed, and we go, is it adaptive? Well, it's in very high frequency in the, looking at the genomics, so it looks like it's selective sweep, and so, wow, that's weird. I wonder what causes it, and then we start looking, and it gets more complex, because we just thought it was a polymorphism within the wolf lineage. It's not. It's actually material that was transferred in. Okay? Great questions, by the way. Still. Okay? How about, yes, go ahead.
The Templeton paper,
Yes.
where there's an overlap between genetic exchangeability and demographic exchangeability. Does it mean that is not a species? We cannot say that they're different species.
So if they're demographically exchangeable and genetically exchangeable largely, he would argue that it's likely that they're at the best something subspecific is what he would argue, applying his species concept. However, if they do not show, I don't know how, see, this is the other part of his that I'm not exactly sure that I agree with. You can't be completely demographically exchangeable. And you know what I mean? I mean, well, or at least in ecological modeling or ecological theory, you can't have the same niche. So really what it amounts to is it's largely demographically exchangeable. But if you're completely demographically exchangeable, then I don't know how you would ever be non-genetically exchangeable. But at least theory-wise. But anyway, yeah, if you're both demographically and genetically exchangeable, you are the same species by his species concept, by applying that. Yes?
So can we say that, as you said about linkage disequilibrium, so is that a limiting force for speciation or causing radiation?
You mean genetic exchange, is it a limiting force or?
Linkage disequilibrium.
Oh. So it's interesting because so linkage disequilibrium. So yeah, it's both. And this is a really cool part of all this. So is linkage disequilibrium, does it promote or retard speciation, basically? And the answer is that as linkage disequilibrium builds up, you have disruptive selection within a population. We can think of it as sympatric speciation. We can talk about it in a few minutes. But so you can end up with two subpopulations that are, with disruptive selection, right? With two different phenotypic spaces that they're occupying in genotypic spaces because you have the linkage disequilibrium. But the interesting thing is then when you have genetic exchange like we do, the outcomes of that would be to reduce linkage disequilibrium initially, right? You disrupt the LD. But then if that introduced material is adaptive, if it increases fitness, many times we would assume it would be neutral or it would decrease fitness like any new mutation. But if you introduce it and it increases fitness, then very quickly you're going to get LD again, linkage disequilibrium. But this case, it's going to be with this new material. And that linkage disequilibrium, if it comes from another organism, so now you have a different, sort of like almost a Wrightian peak that you're on. In fact, that's what it's been argued to be like. So a fitness peak that nobody is occupying. The two parents aren't occupying. Because now you have this third form. Is it going to be a new species? Possibly. Possibly. Or we could consider it, and this is where it gets definitional. I don't think it's trivial, but I think it gets definitional. Are we, for example, what's referred to as a homoploid hybrid species? We're stabilized. We're a single form. The variants, we don't have different species within our human species. We don't have different subspecies. We know we're a hybrid. We're an incredibly admixed unit. Recent, in the last, 45,000 years, we've been hybridizing. We hybridize with everything that, I almost said wouldn't get out of our way, but basically that's it. Well, we're a primate, okay? Guys, we're a primate. I've written books on it. Primates hybridize, okay? They just do, and we are a primate. So we get to the point where when that LD builds up again in us, let's say, and we have locally adapted populations. You guys are locally adapted to your area. Your skin color is more similar to my wife's, darker. Your hair type will be different from mine. I'll be locally adapted to not being out in the sun or I'll get skin cancer kind of thing. And so, when that LD builds up, now are we a homoploid hybrid species because we have all this introgressed material, or are we just an introgressed Homo sapiens? And that's where it gets definitional. But going back to your point, LD can promote and also bring back together when it's reduced, and then promote again. That's why we like measuring LD, which, by the way, coined, I believe, by Dick Lewontin, and it was. And I was talking to him one time, and I said, linkage disequilibrium? Really? And he said, I know, it was the lousiest thing I ever came up with. He said, it was great. He said, the concept in this, I said, it's powerful, absolutely critical for us to know and measure, but the term, linkage, what does that mean? Really, I mean, seriously, I know what he was trying to mean, and anyway, he was very gracious. He didn't hit me. So, and he could have. He's really smart, okay? All right, other questions? This is great, again. Okay, why don't we go ahead and start talking about modes of speciation, okay? What is Ernst Mayer's, how do species form according to Ernst Mayr? We've talked about it some, and you probably know already before we talked about it, but how does he say species have to form? In terms of geographic, what does he tell us? Geographic terminology. So we call it what? Allopatric. Okay. I think this is really important for you to remember. Whoops. Sorry. Just give me a second. I'm a little bit heavy-fisted. If you can't tell, I began my writing of manuscripts and stuff on a typewriter. I know. You don't know what one of those are, do you? All right. This is the cool thing to remember about what came out of the neo-Darwinian synthesis and why many of us have been trying to build this new paradigm and have, I think, successfully built this new paradigm, but it took a bunch of years, took 20 years. That geographic speciation, and what he means by this is allopatric. Okay, that's, his terminology was geographic, but it's allopatric speciation, is the almost exclusive mode of speciation among animals, and most likely the prevailing mode, even in plants, is now quite generally accepted. This was in 1963, and so what he was saying was, we know that plants and animals had to evolve in allopatry, and everybody generally is accepting it. This is one of his all thinking biologists agree with me kinds of statements. Seriously, this is one of the places where he says that too during this book, in this book. So all plants and animals, and I think that's interesting that he says plants there. But in 1992, he was in Harvard still, and he went into the herbarium, and he looked at the herbarium sheets for the plants that occurred in Cambridge, the city that Harvard's in. And he wrote a paper and published it in the American Journal of Botany, which said plants don't hybridize either. And basically, and he said they're really low. He said there's no evidence. I looked at these herbarium sheets and blah, 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 blah. So you need to keep in mind that's what came out, and we'll look at this again in some talks that I'll have about quotes from him. But keep in mind that this is the paradigm that came out of the neo-Darwinian synthesis. Okay, it's very important to keep that in mind because that gives you an idea of why it's been necessary for a number of us to test these assumptions, okay, throughout time and ways we've tested them. We'll talk about that. All right, so allopatric speciation. Why do you think Ernst Mayr reasonably, in some ways, would have argued that it had to be allopatric? That for species to form, there had to be allopatric separation. Why couldn't they be sympatric? And why does it, so let's say we get new mutations, and some of those might lead to divergence from one into two types. What do you mean that it disperses? What is happening there that keeps it from, in that single population, from diverging into two? You're right, but why? What are we talking about? right? And so what we're talking about is simple recombination in meiosis, right? And that breaks up novel genotypes. And this goes back to your question about LD, obviously, linkage disequilibrium. How do we build up linkage disequilibrium if we don't have that, right? If we don't have from one splitting into two geographically, becoming allopatric, and then you can have LD build up, i.e. new genotypes, different linkage disequilibrium between different alleles, building up over time, and allopatry. And you need to keep in mind that this wasn't idiotic, but it had a series of assumptions, not observations. One assumption was that selection was always weak. And the second assumption that really follows on from that is that divergence always or nearly always slow. Okay, so in other words, it takes many, many generations to end up with this that started out the same being two different species. And selection was always very weak. It's a Darwinian kind of model, right? I mean, Darwin argued that evolution took long periods of time with very slight incremental changes over that time. There's another assumption here, and that is, now we're going to read a paper by Meyer where he argues for another kind of model, but it was not something that he really pushed that hard, but we needed to read it because it follows on to something else. But that divergence was also continuous. Not episodic. Okay, not always, but in other words, that you had continuous evolution for a long period of time, continuous buildup of mutations for long periods of time, with selection being incredibly weak, and on average incredibly weak, all across that time period. And so if selection is weak, and divergence is always slow, and divergence is continuous, and you put these back together, well, actually, Monty Slatkin, the rule was, okay, that he came up with, and Landy also amplified on this, one migrant per generation will stop divergence. One migrant per generation between these will homogenize those, okay? With that kind of theory, in case the one migrant per generation obviously came later, Monty Slatkin and Landy worked on that in the 70s and 80s, I guess it was. With that kind of theory, of course they had to be allopatric. But it's not a theory, it's not an observation, I mean, it's an assumption. Because all of these are assumptions. But if these are correct, then this is correct, that you have to have allopatry. And if that's correct, you have to have allopatry, okay? So, allopatric divergence. So did Meyer ever change his ideas? This is important, I think, historically as well as for you guys to understand. So Ernst Mayr, whoops. Let me do something real quick. I want you to be able to see something that's disappeared on me. See if I can do this. Stop. Ding, 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 ding. Hang on, just let me try one more time. Oh, screw it. All right. Sorry, shouldn't have said that. So, this is supposed to be red from here on up. So, Ernst Mayr was asked when he was 100 to comment on the field of evolutionary biology. This was 2000 and some odd, I don't remember when it was, in Science. It was cool. And they asked him why he was so influential in one place. And he said, well, because my ideas were so powerful and correct. But he said also I outlived everybody. So I thought that was finally had a little bit of self-awareness. Here's what he says first, which I absolutely agree with. It would seem justified to assert that so far no revision of the Darwinian paradigm has become necessary as a consequence of the spectacular discoveries of molecular biology. Let me add one caveat: epigenetics. And so we're going to have to really watch for the next 5 years. Some of us were talking about it today. My bet is if I rewrite my book in 5 years, I bet you any amount of money I have a chapter on epigenetics. I mean, I already talk a little bit about epigenetics in there with the allopolyploidy and gene silencing and all this sort of thing in plants. But I'm talking about how influential, impactful has epigenetics been, epigenetic process has been in evolution. Okay? And so it'll be interesting to see. But anyway, I agree. We really don't need to overturn the Darwinian paradigm. But here's where it gets really interesting. And you can sort of see this better. So he claims, remember this is in 2000 and some odd, he claims that they've looked at all these different animals and speciation has been observed to be allopatric, geographical in most of the studied groups. This is where it really gets wonky. No exceptions have been found in birds and mammals where we find good biological species, and speciation in these groups is always allopatric. So I guess, you know, geez Louise, I don't even know what to say. But the thing about it is, what I can say is that a number of evolutionary biologists who worked on animals, particularly birds and mammals, went, what? Are you kidding me? Like Darwin's finches. And so anyway, the bottom line is, though, you need to understand, this was in the journal Science. Okay, it wasn't in the journal of, you know, irreproducible results. Should have been. But the thing about it is that you need to understand that this is still ongoing as a paradigm that Coyne and Orr write speciation books around, and other people may too, that we need to continue to remind ourselves that we actually have data to test these conclusions. We had data then, but we really have data now. And the data are not agreeing with this conclusion, and they sure don't agree with, you know, largely allopatric and bugs and, you know, Lepidoptera and things, and especially plants. But you need to understand that this is still the biological species concept, the allopatric model of speciation is still thought of by those particularly who are interested in speciation, speciation with those assumptions. And what we have to do, and what people are doing repeatedly, is to test these assumptions. Okay? Because the predictions are the same, right? The predictions are, in terms of biodiversity, we will get biodiversity. Those predictions are going to overlap whether these are correct or not, so we need to test these. And that's what lots and lots of folks are doing right now. Not just our lab, obviously, not just our group, but many people around the world are testing those and have been testing those for some time. So anyway, I just wanted to give you, you know, just a little snapshot of it. It's not 1963 where, you know, Ernst Mayr then changed his mind and et cetera. He really didn't change his mind, and many people who followed that allopatric, some people who follow a strict allopatric model are still trying to hold on to that. And what I would say is why? You know, I would ask the question, well, why hold on to it? It's beautiful. I mean, that it's more complex. Now we can ask, do you have to have allopatry part of the time to have biodiversification? I would think you probably do. But you also have to have that admixture. So, you know, it's that sort of thing. Let's celebrate the complexity. That sounds way too much like a Coca-Cola ad, but whatever. Okay. So Ernst Mayr's model. Comments? We have some other papers we can go through. Let me just get rid of that. Pull this one back up. Okay, comments or questions about this? Of that particular model? Yeah.
How easy or difficult is it to have a mechanism that fairly instantly brings about pre or post psychotic isolation within a population?
It's a great question, and that's probably, I mean, let's just talk about it in the context of these. So we're talking about sympatric, right? We're talking about sympatric divergence. And the question, I would argue that the question is still there as to how frequently we have purely sympatric divergence. And really what I mean by that is, does it take a certain life history, whether it's plants or animals, for sympatric divergence or certain kinds of characteristics? Okay, now we're talking about at the diploid level. So let's take a giant step back and say, with Ernst Mayr in his book, that the best example of instantaneous, quote unquote, those were his words, but instantaneous sympatric speciation is allopolyploidization. And we know all angiosperms are allopolyploids, probably all plants. We know we are polyploids, and it's likely, looking at the data, that we're allopolyploids, somewhere in the vertebrate lineage. Okay, so what we can say in terms of that scale, not just looking at neo-allopolyploids, but looking all the way through plants and animals, that it's been an incredibly important sympatric mode of speciation because it's affected everything. Not neo-allopolyploids now, but it's affected for some reason it's very successful. That's another question we could ask. If neo-allopolyploidization reflects how frequent allopolyploidization is in plants, let's say, and we know it's, say, 4% of the time or whatever the value is, why is it 100% of all clades are made up of 100% of all species being allopolyploid? How did it get sorted out like that? Why don't we have 4% and 96%? So it's an interesting question, and there's demographic clade demographies that could, maybe account for it. But anyway, now stepping back into your question, so I'm just saying that actually sympatric diversification, before I say that I wonder how frequent it is incredibly frequent in terms of allopolyploidization, whole genome duplications. Now, what about homoploid, diploid level sympatric divergence? I'm still not convinced that it's not a relatively rare, I'm just talking about across organisms, animals and plants, that it may be relatively rare. Okay, where you have a single population, you have a polymorphism. This is strictly sympatric model, like Saravaya's model. You have a polymorphism arise, not genetic exchange. Polymorphism arises, and then that polymorphism sorts out across that sympatric region temporally, spatially, and you end up with two subgroups that then go on to diverge. I'm not sure if that wouldn't be that restrictive restricted definition of sympatric speciation, which is the one that Ernst Mayr was using. So we need to understand that's the definition of sympatric speciation, really, and what Sarah is talking about and what Guy Bush is talking about. So with that definition, it may be a relatively small proportion of plants and animals that would actually show that, to have the traits that would actually be able to have that kind of speciation event occur. Like these guys worked on, you know, insects with host plant specificities, or parasites that live in certain environments like in our guts or something, you know, where they have a micro niche kind of distribution. So, yeah, how hard is it? Well, what is Sarah, do you guys remember? I mean, what did they have here? What does Sarah argue in her review, the Ugly Duckling review? What are the difficulties here? It goes back to linkage disequilibrium and building up and that sort of thing and sympatry. But it's really overcoming that, right? It's recombination. It's having strong enough selection that you have a distribution. So you have a phenotypic space Okay, we'll get there, I promise. So you have obviously, what we're talking about is having a phenotypic, genotypic space, right? And the question is, can you have strong enough selection to end up with something like that without forcing this to extinction? So that was Maynard Smith's and others, Doug Fatuma and others argued that the strength of selection to end up with, and these have different constellations of alleles that are being held together by LD by natural selection, okay? Probably not by real linkage. So the kind of selection to maintain this, but the kind of selection that would disruptive, or in some cases it's differential. But anyway, should, theoretically, they would argue, should drive it to extinction. Okay, yeah.
Most cases of most papers about adaptive radiation also talk about these kinds of models also.
Adaptive radiations. So, no, not really, okay? But I'm going to add a caveat in a moment, okay? And about introgressive hybridization because of that, some of the observations. But in terms of adaptive radiations, instead of this, normally what adaptive radiations perceive or are perceived as involving is a distribution into many, many niches that are open. So it's not disruptive selection, but rather a release. You have phenotypic and genotypic variability enough that you're released across a landscape of niches that are empty. That's the model. Okay, what I will say is, once again, and we've talked about it before, is it's looking more and more like many of the examples of what we refer to as adaptive radiations are underlain by sympatric events, i.e. introgressive hybridization. But it's still perceived as releasing these organisms across environments. But if it's sexual selection, it could easily be then turned around and is driving these adaptive radiations and they're hybrids, then it's still sympatric. And in that case, it's like a host, a magnificently complex insect plant host kind of thing where you have lots and lots of different hosts. The female is selecting those hosts. So it's getting more complex, but historically, adaptive radiations were not thought of as disruptive selection, maybe directional, that you had a lot of different types that sorted out into the environment, and then directional selection favored certain phenotypes in certain environments.
Do you have empirical evidence for disruptive selection?
Say one more time.
Do you have examples of disruptive selection?
Yeah, I mean, if you look, well, we have experimental examples too, but the disruptive selection, Sarah has detected it in her aphids, okay, in the fields where they have different host plants, and they've also detected it in ragolides. So disruptive selection in terms of having that kind of phenotypic space and losing the center portion of it, yes. But once again, that specific kind of model, she has a number of different examples, though, really pretty examples. So I'm not negating that that kind of model of sympatric speciation divergence is not occurring. I really think it is. It's just I'm not sure frequency-wise whether or not it is plays a major role. And it's probably because of how I was raised as an evolutionary biologist. I was reading Mayer and Doug Fatuma and Monty Slatkin, and I was reading all these people and thought, Maynard Smith, and thought, oh yeah, that makes sense. So sympatric speciation really can't work like that, which is ironic considering what I work on now. But so I'm just not sure what the frequency of the kind of thing Sarah's writing about is. Doesn't make it unimportant, I'm just not sure. But it's very important. And the one thing I will say is the reason you read these is because coming out of the neo-Darwinian synthesis, these kinds of ideas were not supposed to be studied because they were trivial at the best and completely inaccurate at the worst. Okay? If you read the Festschrift, the volume where we celebrated Guy Bush's whatever, I don't remember, we were all out, a bunch of us were out in Asilomar, California, and we gave talks. And he does a little history of his, this is 1969, this is a foundational paper for sympatric speciation, at least in North America, thinking about it. And he was a graduate student. This was his PhD work that came out then. And if you read the little history he did in the back of this volume we produced, Guy tells the story that Guy did his PhD at Harvard. Ernst Mayr was on his committee. He said, Guy says the only reason he got his PhD is because he took his PhD to Ernst Mayr to read. His PhD, of course, was all about sympatric speciation. Took it to Ernst Mayr's house for him to read it, and Ernst Mayr said, I'm leaving tomorrow for Papua New Guinea. I don't have time to read this. Get somebody else for your committee. And Bush said that that's the only reason he ever graduated, was because Ernst Mayr didn't get a chance to just trash his PhD. And then he went to the evolution meetings, the Society for Study of Evolution, as a brand new minted PhD student, to give a talk on this paper and a companion paper in evolution. He gave the talk, and Theodosius Dobzhansky was the moderator of the session that he gave the talk in. Theodosius Dobzhansky also did not believe, quote unquote, in sympatric speciation. He thought it was a bunch of hooey. And so, which is a word that I can't translate into whatever. So Guy Bush said he finished his talk at the end, and he stood there, and you know how it is. This is a contributed paper. And you're standing there, and you're just praying that somebody will ask a question to make your life feel worthwhile. And he said not a single person raised their hand. And Dobzhansky got up, and he said, sympatric speciation. It's like the measles. Everybody catches it, but we all get over it. So I try not to do that to grad students or postdocs. I do it to colleagues. When Hema speaks, I'm going to give her a really hard time. I mean, what the heck? She's got tenure. She doesn't care. But I guess why I'm, telling funny stories is that that's sort of the context of sympatric speciation, of genetic exchange, of all this. Just keep in mind that coming out of the neo-Darwinian synthesis, and I keep mentioning that period really solidified a lot of different things that evolutionary biologists think have to be correct. How do we define species? Well, there's only one way, biological species concept. How do they arise? Allopatrically, every time, okay? Because we know that those assumptions are accurate. So it's historically really important to understand why Sarah Baez said the ugly duckling grows up, turns into a swan. In other words, we can now study to see if sympatric speciation is actually a viable model for whatever we're working on, right? The Lord Howe Island flora, sympatric speciation predominates, right? And that was in Nature, I guess it was, or Science, whichever one, not that long ago. They would never have been able to have published it back here. They wouldn't have been allowed. So I'm just saying that keep that in mind, and also keep your eyes open. What is not allowed to be studied in your particular, when you bring it up, what do people always roll their eyes about? And go, jeez, yeah, right, whatever. You know what I mean? Because actually, later on, you may not want to push against that right now as a graduate student, or undergraduate, whatever you are. Keep it in the back of your mind, because maybe later on, you can actually explore that and you become really well known because, oh, wow, look, they were really smart. Well, you would be. Okay. It's been an hour and a half. Should we take a break now? Okay. Why don't we take a break and we'll come back. Look, we're catching up. So we'll come back and we'll discuss these. Okay, some more modes of speciation. Whoops. That wasn't smart.